Good morning, students. Today we discussed about fatty acids. Biosynthesis consists of cycles of two carbon addition. Fatty acids biosynthesis involves the cyclic condensation of two carbon units derived from acetyl coenzyme A. In plants, fatty acids are synthesized exclusively in the plastids. In animals, fatty acids are synthesized primarily in the cytosol. The enzymes of the biosynthesis pathway are thought to be held together in a complex that is collectively referred to as fatty acid synthase. The complex probably allows the series of reactions to occur more efficiently than it would if the enzymes were physically separated from one another. In addition, the growing acyl chains are covalently bound to a low molecular weight acidic protein called the acyl carrier protein ACP. When conjugated to the acyl carrier protein, an acyl chain is referred to as acyl ACP. The first committed step in the pathway is the synthesis of malonyl coenzyme A from acetyl coenzyme A and CO2 by the enzyme acetyl coenzyme A carboxylase. The tight regulation of acetyl coenzyme A carboxylase appears to control the overall rate of fatty acid synthesis. And Jaworski, the malonyl coenzyme A then reacts with ACP, acyl carrier protein, to yield malonyl ACP in following steps. Like first, in the first step of the cycle, fatty acid synthesis. The acetate group from acetyl coenzyme A is transferred to a specific cysteine of condensing enzyme 3-keto-acyl ACP synthase and then combined with malonyl ACP to form acetoacetyl ACP. In second step, the keto group at carbon 3 is removed by the action of 3 enzymes to form a new acyl chain which is now 4 carbon long. The next step is the 4 carbon fatty acid and other molecule of malonyl ACP then become the new substrates for condensing enzyme, resulting in the addition of another 2 carbon unit to the growing chain. The cycle continues until 16 or 18 carbon have been added. Some 60:0 ACP is released from the fatty acid synthase machinery, but most molecules that are elongated to 18:0 ACP are efficiently converted into 18:1 ACP by a desaturase enzyme. Thus, 16,0 ACP and 18,1 ACP are the major products of fatty acid synthesis in plastids. Fatty acids may undergo further modifications after they are linked with glycerol to form glycerolipids. Additional double bonds are placed in the 16:0 and 18:1 fatty acids by a series of desaturase isozymes. Desaturase isozymes are integral membrane proteins found in the chloroplast and the endoplasmic reticulum (ER). Each desaturase inserts a double bond at a specific position in the fatty acid chain, and the enzyme acts sequentially to produce the final 18:3 and 16:3 products. Glycerolipids are synthesized in the plastids and the endoplasmic reticulum (ER). The fatty acids synthesized in the chloroplast are next used to make glycerolipids of membranes and oil bodies. The first steps of glycerolipid synthesis are two acylation reactions that transfer fatty acids from acyl ACP or acyl coenzyme A to glycerol 3 phosphate to form phosphatidic acid. In this diagram, we will see the compartment of chloroplast and prokaryotic pathway. Fatty acid synthesis and 18:0 ACP desaturase. It forms phosphatidic acid, which converts into diacylglycerol DAG, and it also comes from ER, and then it converts into monogalactosyl diacylglycerol and sulfolipids. These monogalactosyl diacylglycerol is now converted into diacylglycerol also. And in eukaryotic, in ER pathway, phosphatidic acid forms diacylglycerol, which forms phosphatidylcholine, and phosphatidic acid form phosphatidyl inositol and phosphatidylglycerol, and DAG is also formed phosphatidyl ethanol amine. The action of a specific phosphatase produces diacylglycerol from phosphatidic acid. Phosphatidic acid can also be converted directly into phosphatidyl inositol or phosphatidylglycerol. DAG can give rise to phosphatidyl ethanol amine or phosphatidylcholine. The localization of enzymes of glycerolipid synthesis reveals a complex and highly regulated interaction between the chloroplast, where fatty acids are synthesized. The other membrane systems of the cell. Two pathways referred to as the prokaryotic pathway or eukaryotic pathway. In chloroplast, the prokaryotic pathway utilizes the 16:0 and 18:1 ACP product of chloroplast fatty acid synthesis to synthesize phosphatidic acid and its derivatives. Alternatively, the fatty acids may be exported to the cytoplasm as coesters. In the cytoplasm, the eukaryotic pathway uses a separate set of acyl transferases in the ER to incorporate the fatty acids into phosphatidic acid and its derivatives. In some higher plants, including Arabidopsis and spinach, the two pathways contribute almost equally to chloroplast lipid synthesis. In many other organisms, however, phosphatidylglycerol is the only product of the prokaryotic pathway, and the remaining chloroplast lipids are synthesized entirely by the eukaryotic pathway. The biochemistry of triacylglycerol synthesis in oilseeds is generally the same as described for the glycerolipids. 16:0 ACP and 18:1 ACP are synthesized in the plastids of the cell and are exported as a coenzyme A thioesters for incorporation into the DAG in the endoplasmic reticulum. So today we discussed about biosynthesis of fatty acids in plants by two methods. In chloroplast, it's a prokaryotic method, and in the cytoplasm, it is called eukaryotic pathways method. So it's finished. Thank you, students.